Today's video is on haemoglobin. So haemoglobin is a protein with a quaternary structure, and we'll see what this means in a minute. And haemoglobin is used to effectively uptake oxygen and offload it in different conditions. So it wants to take up oxygen at the lungs, so where there is a high oxygen saturation, and offloads oxygen at respiring tissues in a low oxygen saturation. So that's what this protein haemoglobin does. It uptakes oxygen in areas of high oxygen saturation and offloads oxygen at respiring tissues where there's low oxygen. So what's the structure of this protein, this haemoglobin? So we said that it has a quaternary structure, and this means that it has multiple polypeptides and prosthetic groups. And so polypeptides are chains of amino acids, so they're protein chains, and prosthetic groups are non-amino acid, non-protein groups associated within the structure of the protein. So our haemoglobin has 4 polypeptide chains. It has 2 alpha and 2 beta chains. So it has these 4 subunits, 2 alpha and 2 beta. And each of these 4 subunits have a heme group. And each heme group has an iron ion. And it's this iron ion, this Fe2+ ion, that can combine with the oxygen molecule. And so, if each polypeptide, each subunit has 1 heme group, and each heme group has 1 iron ion, and this iron ion can bind to an oxygen molecule, then one haemoglobin protein can carry a maximum of 4 oxygens. So haemoglobin's function, we had a quick look at it, so let's go into a bit of more detail. So for effective functioning, haemoglobin must readily associate with oxygen, and associate with oxygen means uptake of oxygen. So it must readily associate with oxygen at the exchange surface. So this means that it must readily take up oxygen at the lungs, because the lungs is our gas exchange surface in a human. And it must readily dissociate with oxygen, so release oxygen, at respiring tissues. So it must give up the oxygen at respiring tissues, so this oxygen can be used in aerobic respiration. And in order to do this, our haemoglobin must change its affinity to oxygen. So affinity is how attracted the haemoglobin is to oxygen. And so in order for it to carry out these functions, in order for it to associate and dissociate readily in different conditions, the haemoglobin must change how attracted to oxygen it is. And so these affinities, so this affinity to oxygen, changes under different conditions. So because the conditions are different in the lungs than in our respiring tissues, the affinity can change. And that allows for this effective functioning. So changing affinity. So how does haemoglobin change affinity, and why does it change affinity? So for association of oxygen at the lungs, so uptake of oxygen at the lungs. So at the lungs there are low levels of carbon dioxide, and this is because the carbon dioxide is diffusing into the alveoli to be forced back out into the atmosphere. And there are high levels of oxygen, and this is because the oxygen is diffusing from the alveoli into the blood for circulation. And so the affinity is high. So if there are low levels of carbon dioxide and high levels of oxygen, the affinity to oxygen of the haemoglobin will be high. So it will cause association of oxygen. It will cause the haemoglobin to uptake more oxygen. And so for dissociation of oxygen at respiring tissues, the levels of carbon dioxide are high, and this is because the respiring tissues have been conducting respiration, aerobic respiration, so they have been producing carbon dioxide. And the levels of oxygen will be low, and this is because they've been using the oxygen in aerobic respiration to produce this carbon dioxide. And in these conditions, the affinity of hemoglobin to oxygen is low. So high levels of carbon dioxide, low levels of oxygen decreases the affinity to oxygen, so oxygen is dissociated from hemoglobin. It is released to the respiring tissues. And so now we're going to look at these oxygen dissociation curves. So what do these show? So this graph will show the relationship of the saturation of hemoglobin. And so saturation of hemoglobin is how many oxygen molecules the hemoglobin is bonded to. So at 100%, this is 4. At 25%, it's 1 oxygen. At 50%, it is 2 oxygens. So this percentage saturation is just how many out of the maximum 4 oxygen molecules is this hemoglobin bonded to. And this is plotted against partial pressure of oxygen. And the partial pressure of oxygen is how much oxygen is present. So this graph shows the relationship between the saturation of hemoglobin and the partial pressure of oxygen. So how the saturation of hemoglobin changes as the concentration of oxygen changes. So we can see as the partial pressure of oxygen increases, this means there's more oxygen, the hemoglobin uptakes more oxygen. So in higher partial pressures of oxygen, the saturation of hemoglobin is higher. And we saw this in the previous slide. So for example, at the lungs, there is a higher partial pressure of oxygen, and so it will uptake more oxygen. So in higher partial pressures, more oxygen, hemoglobin will uptake, it will associate with oxygen. And as partial pressure decreases, so there's less oxygen, the hemoglobin will release more oxygen. And we saw this in the previous slide as well. So at respiring tissues, there are lower partial pressures of oxygen, so that the affinity is lower. And so this means it will release more oxygen. So the hemoglobin saturation will be lower because it's holding on to less of the oxygen molecules and dissociating more of them. It's releasing more of the oxygen. So now moving on to the shape of the curve. So we say that this is a sigmoidal shape, but why does the hemoglobin saturation curve take this shape? So the shape of the hemoglobin molecule, our protein, is closely united. So all of the 4 polypeptide chains are quite close together, and this makes the binding of the first oxygen molecule very difficult, because it was difficult to get to one of the ions to bind. So because they're so close together, this oxygen molecule, this first one, finds it difficult to bind and collide with one of the ions in the heme groups. And because it's difficult for this first molecule to bind, at low oxygen concentrations, little oxygen binds, so the curve is shallow because it is difficult for this first oxygen molecule to bind. And so we know 25% hemoglobin saturation will be the first of the 4 oxygen molecules to bind. And so up to that point, the curve is very shallow because it's difficult for it to bind. So it doesn't bind very quickly. So it takes a lot of this low oxygen concentration for it to bind. And the binding of the first oxygen molecule changes the structure of the hemoglobin. And so the binding of this oxygen molecule will cause a structural change within the hemoglobin, and this will open up the other binding sites more. So this closely united quaternary structure will open up slightly when the first oxygen molecule binds, exposing these binding sites. And so it takes a smaller increase in partial pressure for the second and third oxygen molecules to bind, so the curve steepens. So the second and third oxygen molecules are easier to bind, so the curve is steeper, and this is because the binding sites, these ions, have been exposed more, so it's easier for them to collide and bind with oxygen molecules. And so it's a nice steeper part because it happens more quickly than this first one. and the fourth binding site is harder to fill, and this is due to the probability of the oxygen molecule colliding with the empty ion. So because we've now filled 3, there's a lower probability that the oxygen molecule will collide with the correct empty ion. So the curve shallows again because the probability of it binding to that binding site specifically decreases. And so that's why we have this sigmoidal shape, because the ability of the hemoglobin to bind to the 4 oxygen molecules changes, the probability of it happening changes, so they require different partial pressures and different times of these partial pressures in order to get full 100% saturation. So what are the key words that we need to know with that process? So there's allostery. So this is when the binding of a molecule changes the shape of a protein, changing its affinity to a certain substance. So we saw this in the previous slide because hemoglobin shows allosteric properties, because the binding of the first oxygen molecule changes the shape of the protein, opening up the binding sites, changing its affinity to oxygen, because it's more likely to bind with this oxygen, it's more attracted to oxygen, because these binding sites have opened up, it's more likely to bind and collide with the oxygen. And then there is positive cooperativity. So this is when one action at a binding site makes the other binding site more likely to be filled. So this means the binding of the first oxygen molecule makes it easier for the second and third oxygen molecule to be binded. So this positive cooperativity, so one action makes the action of the others easier. It assists the function of hemoglobin. So what happens when we shift the curve? So we saw this sigmoidal shape in our hemoglobin saturation curves, but what happens if it moves slightly to the left or to the right? So the further to the right our saturation curve means the lower the affinity. So the lower, the less attracted to oxygen the hemoglobin is. So this is because at the same partial pressure of oxygen, the hemoglobin is less saturated. So let me explain this. So if this is normal hemoglobin, and then this is shifted to the right. So let's say at a 40 partial pressure of oxygen, if we go up, at the same partial pressure, at the same 40, our curve that is shifted to the right is less saturated than the normal hemoglobin. And so we say, because at the same partial pressure it's less saturated, this means that it has a lower affinity to oxygen, because at the same partial pressure it uptakes less oxygen when it's shifted to the right. And so the further to the left means the higher the affinity. So at the same partial pressure of oxygen, the hemoglobin is more saturated. So this means at the same partial pressure, it will uptake more oxygen. So it has a higher affinity. So the further to the left, the higher affinity, because it will uptake more oxygen than the others. And further to the right, the lower affinity, because it uptakes less oxygen than the others. And so we want to have a look at the Bohr effect. So the higher the concentration of carbon dioxide, the more dissolves to form carbonic acid. So carbon dioxide dissolves in our blood to form carbonic acid. And the more carbonic acid, it means the more we lower the pH of our blood, because pH is a measure of acidity, and the lower the pH, the more acidic. So if we have more carbon dioxide, we have more carbonic acid, so we lower the pH more. So the higher the concentration of carbon dioxide, the lower the pH. And so the lowering of the pH shifts our hemoglobin saturation curve to the right. And as we saw on the last slide, shifting it to the right lowers the affinity. So the less attracted to oxygen the hemoglobin is. So the hemoglobin is less attracted to the oxygen. And so this is why hemoglobin can release oxygen at respiring tissues, because at respiring tissues there is a higher presence of carbon dioxide, and so if there's higher concentrations of carbon dioxide at these respiring tissues, it will lower the pH more, and the more low the pH is, this lowers the hemoglobin's affinity because the curve shifts to the right. So the higher concentration of carbon dioxide shifts the hemoglobin saturation curve to the right. It lowers the affinity, so it releases more oxygen to respiring tissues. And it does this, the pH lowering can change the hemoglobin's affinity because the change in pH changes the shape of the hemoglobin. It changes the structure of these 4 polypeptides. And so this changing in shape changes the affinity, and it will release more of its oxygen to the respiring tissues for aerobic respiration. And lastly, we'll look at an example that is heavily brought up in exam questions. So we're going to use mouse versus human hemoglobin saturation curves. So why is the hemoglobin saturation curve different between a human and a mouse? So the mouse, we can see, has a lower affinity to oxygen because the curve is shifted more to the right. So at the same partial pressure of oxygen, it has a lower hemoglobin saturation. So we know that a mouse has a higher and larger surface area to volume ratio than a human. So this mouse has a larger surface area to volume ratio. So this means that the mouse will lose heat more rapidly, and so it needs a higher metabolic rate to upkeep their body temperature because they're quickly losing their body heat because of this large surface area to volume ratio. And so the mouse needs a lower affinity to oxygen because this means it will release more oxygen at respiring tissues. So the lower the affinity, the more shifted to the right their hemoglobin saturation curve means they release more oxygen at their respiring tissues. And so the more oxygen their respiring tissues get means the more aerobic respiration that their tissues can carry out, means the more energy released via this aerobic respiration, so their higher metabolism is maintained, so they can keep up their body temperature. And so this is why their curve is shifted to the right, because they want to lower their affinity to oxygen so they release more oxygen at their respiring tissues.